Hello guys. So now let us start with biochemistry. Just by hearing the name biochemistry or just by seeing the topic, we all would get scared of the vastness of biochemistry, right? Since our first year, this has been a big headache. But just bear with me, with just 3 pages, I'm gonna clear off your entire biochemistry and I'm gonna make it so easy and interesting. You'll fall in love with the subject of biochemistry. Okay, this is very interesting with respect to all the biochemical stuff that goes on inside our cells, all the hormonal things, etc. So it is very interesting. Just bear with me, and you'll be done with biochemistry for your entrance exams. Okay, so starting off, we'll go with the nuclear biochemistry. So in nuclear biochemistry, the information that you need to have is the structure of DNA and what exactly the bonds are with respect to DNA. Okay, so between A and T. Okay, so remember this in that way only. Okay, between A and T, A and U, and G and C. So this bond is gonna be H bond. Okay, hydrogen bond. Remember in that way, instead of between nucleotide, between nucleoside, what is the bond? In that instead of that, it is better you learn that between A and T, the direct bonds will be H bond, and between the adjacent nucleotides, it is going to be the phosphodiester bond. This is a DNA structure, right? So in the DNA structure, between A and T, it's going to be the H bond, whereas between adjacent nucleotide, it is going to be the phosphodiester bond. That's it. Then the other important stuff I want you guys to remember is histone is the protein that binds all the DNA material. Okay, all the DNA material together will be binded by histone proteins. Okay, that's it. Then coming to salvage pathway enzyme defects. Okay, so what is this? So there are two pathways through which the sugars of the DNA will be produced. Okay, so one is de novo pathway. Then the other one is salvage pathway. So through de novo pathway, this is a fresh start for the sugar. Okay. Then salvage pathway, with the already existing sugar, it is gonna recycle and produce the new DNA materials. Okay. So if already existing materials are there, then it is gonna be the salvage pathway. It is gonna follow. Okay. So in salvage pathway, need not learn about all the pathways and all. Only the pathways that I tell you to learn is what you're gonna learn. Okay. So all the pathways and all, there are hundreds of pathways. You need not learn everything of it. Okay. Only the pathways I'll tell you, and I'll teach you all the pathways that is very required. And for other stuff, I'll tell you only what is required in that particular pathway. For example, salvage pathway, you need only the enzyme defects that are important. Okay, so that is why you need not learn the entire pathway with it. Okay, in salvage pathway there are two enzyme defects. Okay, one is ADA deficiency and the other one is HGPRT deficiency. Okay, if ADA deficiency happens, this forms a pathology called SCID. Okay, then HGPRT deficiency. So in HPRT deficiency, when the PRP increases, the uric acid also increases. Okay, so normally when there is HGPRT, the PRP will be reduced. Okay, so there won't be any increase in uric acid. But when HGPRT has deficiency is there, so it is going to lead to increase in PRPP, which will lead to uric acid increasing. Okay, so which leads to a disorder called Lesch-Nyhan syndrome. Okay, you would have definitely heard these two names. Okay, SCID, which is severe combined immunodeficiency, and also Lesch-Nyhan syndrome, but you wouldn't have known like what exactly those deficiencies, defects or the pathologies are. So what happens is, in the salvage pathway, I told you, right, Salvage pathway is where the already existing sugars will be forming the DNA material. Like recycling, it is nothing but recycling. Okay, in that pathway, if ADA deficiency is there, then ADA and HGPRT are two enzymes that are required in that pathway. Okay, if ADA deficiency is there, it is going to go to severe combined immunodeficiency, whereas HGPRT is also an enzyme in that pathway. If the defect, if there is a deficiency in that, so it is going to increase the uric acid, so which is going to lead to Lesch-Nyhan syndrome. So Lesch-Nyhan syndrome is gonna have the features of hyperuricemia, because uric acid is increased. Okay, then chorioathetosis can be seen, then mutilating behaviors can be seen. These are the three main symptoms the HGPRT deficiency, or exactly to be precise, Lesch-Nyhan syndrome will be having. Then coming on to DNA damage and to DNA repair. Okay, now that we are done with what how the DNA is gonna look and what the salvage pathway deficiencies, enzyme deficiencies will cause, let us go on to DNA damage and repair. The two main things which, if there is a problem, will lead to cancer. Just think in that way. So that is why they are very interested in asking questions with respect to this topic. Okay, so DNA damage and DNA repair. If there is a DNA damage, it is going to go into mutation. If all the mutations won't turn into cancer, how does it happen? From DNA repair. If DNA repair is there, then it is gonna like put off all the mutations, right? So that is how all the DNA damage won't cause the cancer. Okay, so let us learn both of the stuff. So DNA damage, if there is many mutations, the only mutation that you need to focus on is point mutation. Okay, in point mutation there are three types: missense, nonsense, and silent. Okay, missense, nonsense, and silent. When, what is missense? When there is a amino acid difference, the protein gets altered. Okay, when there is a amino acid, for example, for one there was supposed to be one amino acid. If it was replaced by a different amino acid, then the protein alteration will happen. Okay, so this is called point mutation, missense. Okay, if there is nonsense, it is going to be stop codon. Okay, if there is a nonsense mutation, then it is gonna happen, what is gonna happen is it will become a stop codon. Okay, there is a protein synthesis happening. See, think of it in this way: there is a protein synthesis happening from a DNA, and suddenly a amino acid, a different amino acid comes up here. So what happens? The entire protein will get differentiated, right? The formation of protein will entirely change because the that particular amino acid was different. Okay, then nonsense protein is nothing but, like, for example, one of the mutation happens is nonsense. Okay, it is not, it was not supposed to happen. So nonsense, what happens is it is a stop codon. The protein synthesis is happening. Suddenly, if there is a stop codon is the codon that tells, it is like a software that tells the protein production to stop. Okay, suddenly if a stop codon comes in between the protein synthesis, then the protein production will stop. This is known as nonsense mutation. Then in silent mutation, the mutation has happened, there was a DNA damage, but it did not lead to any change, no change. Okay, then it is gonna be silent mutation. Okay, silent point mutation. That's it. These are the three types of point mutations: missense, which is gonna alter the protein; nonsense will stop the protein synthesis; whereas silent protein, there won't be any change with the protein production. That's it. Okay, then these mutations are happening. How will the DNA repair? Okay, so let us see. So DNA can be repaired as double strand and also by single strand. Okay, so if the double strand, both the strands, the DNA is a double stranded, right? So if both the strands are damaged somewhere, then double strand repair will happen. If suppose only single strand is affected, then the single strand repair will happen. Okay, so in double strand, there is non-homologous repair and homologous repair. Whereas in single strand, there is nucleotide excision repair, base excision repair, and mismatch repair. Okay, so I want you guys to learn the examples because this is a very important previous year questions. In every year, they have asked you one question from DNA repair. Okay, so if you know the example, you'll definitely be able to solve this. Non-homologous repair example is SCID. We discussed just above, right? Severe combined immunodeficiency disorder. This happens because of non-homologous repair defect. See, these things happen when there is a defect in repair. It was supposed to repair, but if there is no defect, I mean, if there is a defect in repair mechanism of the DNA, then these diseases will happen. When there is a non-homologous DNA repair defect, then SCID, and in CRISPR gene mechanism also, this is what we are using it. Okay, non-homologous end joining repair. Okay, end joining repair defect is what we are using it as a technology. See, this is the technology that we are using, so that is why these two examples are important. Whereas in homologous end joining repair, breast cancer is the example. Then coming to single strand repair, I told you, right, the single strand is there. So if there is a damage, then there are three types of repair, that is nucleotide excision, base excision, mismatch repair is there. So if there is a defect in these repair mechanism, then it's going to lead to nucleotide excision repair. If it is defect, xeroderma pigmentosa. Note down, this is the most of this is the question that was most commonly asked. Okay, nucleotide excision repair is xeroderma pigmentosa. Then coming to base excision repair, if it is defect, then it is gonna form MAP. Remember it that way only, MAP, that's it. Okay, need not learn the abbreviations of it. Then mismatch repair, if there is a defect, then it is gonna cause HNPCC. Okay, so please note this down for your sticky note. Okay, that's it. We got to know the entire nuclear biochemistry. This is all and everything that you are supposed to know with respect to biochemistry, because they have been focusing more on what are the clinical aspects with respect to nuclear biochemistry. You need not learn the entire DNA chapter or everything about the DNA chapter, and you will be done with the entire nuclear biochemistry if you study only these topics. Okay, one or two other things that you will be missed will be covered, like if you are solving your any of the question banks and all, just add to these notes. You can't add each and everything, right? One or two points would be missed. For example, I have missed a fish. Okay, so these are all fish thing and all have missed. So please add those things when you see a question about it. Okay, that's it, and your entire nuclear biochemistry is done. Okay, moving on, the protein synthesis. Okay, so now the DNA has been formed, and protein synthesis is what DNA is nothing but it gives the code for the protein synthesis, right? So protein synthesis will be affected, or protein synthesis has four steps. Okay, initiation, elongation, termination, and post-translational modification. Okay, these are the four steps in protein synthesis. So initiation happens. Okay, a DNA is there, a mRNA is there. mRNA, by reading the mRNA, this is like a software. Okay, so software tells you what protein it should produce. Okay, so ribosome will be there, and these ribosomes start producing a protein. Okay, what does this protein do? We learned about this in general physiology, right? So these are the proteins that will go and attach to a receptor, the particular receptor, and causes that particular metabolism. Okay, so that is why protein production is important. So this particular protein production that we discussed about has four steps: initiation, elongation, termination, and post-translational modification. The two things I want you guys to remember is, in elongation there is elongation factor. Okay, this is the particular thing that is affected by diphtheria. You know, diphtheria toxin, the DPT vaccine that we give and all. So if there is a infection of diphtheria, this is exactly where it will go and act. Okay, this is very important. Please remember this elongation factor. Okay, in the protein synthesis is where it is going and affecting it. Okay, then in post transitional modification there are two things. One is N and C cap removal, then phosphorylation. Okay, not much of an importance, but this elongation factor thing is very very important. Please remember this. Okay, I'm gonna repeat this again in microbiology, but for now you please remember where I'm exactly talking it about. Okay, that's it. Now moving on further, let us go to cellular biochemistry. So the first topic will be cell cycle. Okay. So in cellular biochemistry, I'll tell you only what is very very important and focus only on those things. Okay. In cell cycle, they have asked zero questions with respect to cell cycle. Okay, so need not learn everything, just I want you to know the clinical significance with the cell cycle. Okay, so instead of learning only the important stuff, I want you guys to learn like what is the clinical significance of this cycle. Okay, so there are G1 phase, S phase, G2 phase and M phase, right? So these are the four phases that we all know. Okay, there is also one more phase called G0 phase. So let us learn what the G0 phase is and why it is very important. Okay, so in G1 phase, when the cell cycle starts, so there is a gene called P53 gene, RB gene. Okay, what it does is it checks if there is any damage. Okay, before the cycle proceeds further. Okay, during the cell cycle, so P53 gene and RB gene checks like whether if there is any damage in the DNA. So this is all DNA cycle, right? So if there is any damage, these two genes, these are like checkpoints. Okay, so checkpoints will check, and if there is any damage, this will send to G0 phase. Okay, then on it will go. If there is a damage, then it will go and form the apoptosis. Okay, I'll tell you what is apoptosis on all when we are discussing about general pathology. But for now, you remember that G1 phase, these two genes are there. These are like checkpoints. And if there is a defect, then it will send back to G0 phase. Okay. It will tell you are not proper. You go back. Okay. That's it. It will not allow the further transition. When P53 gene and RB gene is affected, that is when the pathology of cancers will come. We'll discuss more about this in neoplasia. Okay, for now you remember this is the cycle. Okay, that's it. So that is where the G0 will come in. Okay, that's it. Then with respect to organelles of the cell, so remember, right, in general physiology we learnt about the cell membrane, then its proteins, etc., and we learnt about the cell skeleton, that is all the cytoskeletons and all. So we did not learn about the organelles, that is mitochondria, nucleus and Golgi complex and all these peroxisomes. All these things we did not learn. So let us learn what are very important only. Okay, you know mitochondria is powerhouse of cell, that you have learnt from your first standard also, you know that. Okay, but only what is important we'll learn here. The organelles that are important and you should be focusing on is the peroxisomes. Okay, so peroxisomes, all the other organelles, you know their functions also. There are not much clinical significance if there is a defect in it, but this peroxisome is very much important because this peroxisome is like a dustbin clearance. Okay, so it clears the waste materials. Okay, just like lysosomes, it clears off the waste materials, and also it is very very important in clearing the fatty acid accumulation and all. So that is where the pathology comes in if this peroxisomes are not proper. So that is why it is very important to learn about peroxisomes. Okay, so what are the things about, or what are the pathologies? Let us see. Okay, suppose if the peroxisomes is absent, that it is going to go and lead to two pathologies, that is Refsum's disease and Zellweger syndrome. Okay, if you are attending many of the question banks, you would have definitely seen some of the questions with respect to these two pathologies. Okay, let us see what is it. So if there is alpha oxidation of fatty acid deficit, I'll explain you what is alpha oxidation, beta oxidation, all these things, and long chain fatty acids, etc. When I'm telling you about biochemistry of metabolism of fat, okay, of fatty acid metabolism, I'll teach you. Right now I'll be teaching you, but just remember, if there is alpha oxidation defect, then it is gonna go and form refsum disease. Whereas if there is long chain fatty acid accumulation, if there is a beta oxidation defect, then it is gonna form Zellweger syndrome. How is it going to present? If there is refsum syndrome, then it is going to present with scaly skin and short toes. Okay, please remember this. Please make sure it is in your sticky note. Okay, these two things, refsum disease and Zellweger syndrome. So the two main clinical features of this refsum disease is scaly skin and short toes. Then when it comes to Zellweger syndrome, there is going to be hypotonia, seizures, and craniofacial dysmorphia. Okay, these are the presentation that the Zellweger syndrome might appear with. Okay, that's it with respect to paroxysms. Then moving on, we discussed about cytoskeleton, right? So I was telling you, I'll discuss about the pathologies of cytoskeleton when I come to biochemistry, right? So let us discuss those pathologies right now. So there are three cytoskeletons, right? Microtubules, microfilaments, and intermediate filaments. I told you, right? Microtubules and microfilaments are the dynamic, whereas the intermediate filament is static. Okay, that's it. Then when it comes to the defect or deficiency with respect to cytoskeleton, let us see what are the pathologies that it might appear with. The first thing you should be focusing on is ciliary dyskinesia. What is this? So basically, I told you, right, this is a cell, and suppose the flagella, cilia, and all is there. Okay, so all the cilia, etc., should also be having skeleton, right? We have our axial body, axial skeleton is there inside our body, vertebra, skull, and all is there. So other than that, we have also have limbs, right? Limbs, so there also the bones are there, radius, ulna, femur, all these things are there, right? In the same way, so cilia will also be having cytoskeleton. Okay, if there is cytoskeleton defect in cilia, then it is gonna have something called ciliary dyskinesia. The cilia won't be beating. Cilia should be beating, right? If the cilia is not beating, then it is gonna cause ciliary dyskinesia. Kinesia meaning movement. Dyskinesia is akinesis. Okay, that's it. So if there is defect in this, the cilia is getting affected, so then it is gonna form a disease known as Carter-Jenner syndrome. Okay, Carter-Jenner syndrome. This definitely you would have heard this name a lot of times. Okay, so what is the triad of Carter-Jenner syndrome? It is gonna come with situs invertus, sinusitis, and infertility, because situs invertus, cytoskeleton is the most important thing that is needed for all the like embryological genesis and embryological rotation and all, right? So when there is a defect in cardiac rotation, it is gonna form situs invertus, because situs invertus is heart was supposed to be on the left side. If it is on the right side, it is gonna form situs invertus. Okay, so that's it. So situs invertus is one of the major pathognomic of Carter-Jenner syndrome. Sinusitis, because cilia is not beating, the mucus clearance is not there. Whereas infertility, because cilia is affected, the vas deferens and all these things aren't able to move the sperm or semen forward. So that is why the infertility is there. Okay, that's it. So I wanted to learn about Young's syndrome here itself, because it is same like Carter-Jenner syndrome, same, but there is no situs invertus, meaning sinusitis is there, infertility is there. Suddenly you will be thinking about Carter-Jenner syndrome, but no, it is gonna be the Young's syndrome, because only if situs invertus is there, we are gonna tell it as Carter-Jenner syndrome, or else it is gonna be Young's syndrome. Okay, that's it. Then moving down, let us discuss about collagen types and its defects. Okay, I told you, right, collagen is very important in our body. I was telling about this collagen in orthopedics, in dermatology also. So let us see what are the types of collagen and what are the defects that we see in if there is a collagen defect. Okay, so there are 4 types of collagen: type 1, type 2, type 3 and type 4. Okay, remember in this way: 1, bone. Okay, 1 is bone, 2 is cartilage, 3 is retricular, 4 is floor. Okay, in that way, if you remember, it is gonna be easy. So collagen type 1 will be in bones and tendons, tendon 1. Okay, then collagen type 2 will be in cartilage, type 3 will be in the retricular formation. For example, it is gonna be in the dermis. Okay, then floor, basement membrane. Type 4 collagen will be in the basement membrane. What is basement membrane? You know that, right? In glomerulus there will be basement membrane. In many other systems, like for example in eye and all these things, we will get to see basement membrane. Wherever you hear the word basement membrane, just next moment itself you should be thinking about, oh okay, type 4 collagen is what they are speaking about. Okay, in this way there are 4 types of collagen, and I've told you the examples of those where exactly these collagen are present in. Okay, then moving down, let us see about defects of collagen. Defect of collagen, first defect we already discussed in orthopedics, that is osteogenesis imperfecta. Okay, collagen defect is there. Bone formation, 1 collagen 1 defect is there, so that is why brittle bone, meaning so for example, there is a long bone, we'll be seeing cracks and fractures in between that entire bone. Okay, so this is known as osteogenesis imperfecta. Then comes another thing called Ehlers-Danlos syndrome. Ehlers-Danlos syndrome is where there is mobile joints. Okay, you would have seen in certain circus people and all, or some of your friends also might be having. This is where they will be able to flex their fingers too much. They'll be they can move their skin too much. Okay, so if you would have seen one piece, Luffy will be having this the exact thing only. Okay, Luffy after eating that fruit will be having this exact thing, that is hypermobile joint. Okay, you know that, right? He can extend his arms, his face, skin and all. So when you think of Ehlers-Danlos syndrome, even in your clinical cases, just think of Luffy from One Piece. Okay, that's it, hypermobile joints. Then when it comes to the other defect of collagen, it is gonna be Menkes disease, because Menkes disease, this definitely we all will confuse with Wilson's disease. Okay, I'll tell you why. Okay, Wilson's disease. Wait, okay, what is Wilson's disease also I'll be telling you. Okay, Menkes disease, this is because of ATP7A gene. Okay, ATP7A gene defect is there, then it is gonna be Menkes disease. If it is ATP7B gene, it is gonna be Wilson's disease. Okay, then there is impaired copper absorption. Okay, in Menkes disease it is going to be impaired copper absorption, whereas in Wilson's disease there is copper transport defect. Okay, transport, when the transport of copper is affected, then it is going to be Wilson's disease, which is ATP7B. Whereas ATP7A gene defect is there, then it is going to be impaired copper absorption. Okay, that's it. Please do not confuse between Menkes disease and Wilson's disease. Just wait for a second, think about whether it is Menkes disease or Wilson's disease that they are talking about. We'll discuss about Wilson's disease in the later chapters. Okay, then Let us also discuss about elastin here itself. Okay, elastin is a stretchy protein. Okay, the reason why I put up here is we should not confuse this with Ehlers-Danlos syndrome. We should not confuse elastin defect with respect to Ehlers-Danlos syndrome. That is why I've put up here. Okay, so if there is elastin defect, elastin is just another collagen. Okay, so collagen is there, elastin is there. Think it in that way. Elastin is also a connective tissue. Okay, just like collagen, it is also a connective tissue. If there is a defect in elastin, it is gonna lead to Marfan's syndrome. Okay, you would have definitely heard Marfan syndrome, and if you are seeing many of the medical dramas, pits, then many other house MD and all, you would have definitely come across Marfans, where there will be long hands and fingers will be there. Suddenly some emergency will come and aortic dissection, suddenly they'll, just by seeing their hand itself, they'll diagnose, right? In airport and all, some fellow will fall, and suddenly by seeing his finger, the doctors will be telling, oh okay, he's definitely having aortic dissection, because he's having a long face, long hands and all. So that is the thing. Is what about Marfan syndrome. Okay, so the main defect in Marfan syndrome is elastin, but the main defect is fibrillin 1. Okay, if in option given, whether it is in the option, they will definitely give Marfans, and they'll be asking you the specific defect. Okay, so if the specific defect is asked, then it is going to be fibrillin 1. Fibrillin 1 is what you should be marking. It is not elastin, it should be fibrillin 1 defect that you should be marking. Okay, I had seen some of the questions also in certain question banks where elastin also was given and the fibrillin 1 was also given, but it is fibrillin 1 that we should be marking. Okay, it is same elastin defect only, but to be specific, it is fibrillin 1 defect. Okay, so then the clinical features I told you, right? Long slender hands and fingers. Then aortic dissection, we'll discuss more about aortic dissection and all when we go to cardiology. Then one of the major thing that I want you guys to remember with respect to Marfan's is there is a lens fanning out. Okay, there is lens subluxation. Here the lens will be fanning outside. Okay, fan is out. Okay, Marfan is out. Okay, because there is also a defect called homocystinuria, which we'll be discussing further down in biochemistry. There is homocystinuria where the lens subluxation is there. Okay, so lens subluxation is there. So we will be like confused between homocystinuria and Marfan's, right? In both of them, lens subluxation is there. So in Marfan's, it is gonna be above and outwards, whereas homocystinuria, it is gonna be downwards. Okay, so this is what I want you guys to keep in mind about. Okay, that's it. So now we are done with the cytoskeleton, collagen defect and all these things. Okay, so let us learn a little bit about genetics and genetic disorders. Okay, now disorders of gene imprinting. Okay, we'll start with disorders of gene imprinting. So here there are two kinds of pathologies, that is Prader-Willi syndrome and another one is Angelman syndrome. This has two very similar type of pathologies. That is why we'll discuss the gene imprinting disorders here itself. Okay, so Prader-Willi syndrome is where the maternal genes are silenced and paternal gene is deleted. Okay, so suppose a chromosome, maternal gene is there and the paternal genes are there. Okay, so maternal gene is silenced. Okay, so already one disorder was there. Suppose if the paternal gene in that particular individual also gets deleted, then if a disorder occurs, then it is going to be Prader-Willi syndrome. P for P. Okay, father, paternal. Okay, so if P is deleted, remember not about silence, remember it is deletion. Okay, so when paternal gene is deleted, it is going to be Prader-Willi syndrome, whereas if mothers, okay, this is vice versa with respect to Angelman syndrome, where paternal gene is silenced, but if the maternal gene is deleted, then it is gonna be Angelman syndrome. Angelman, M for mother. Okay. If Angelman M is deleted, then it is gonna be Angelman syndrome. So how will both of these present with? Prader-Willi syndrome will present with obesity and hyperphagia. Obesity is there, meaning hyperphagia, too much eating is there. Okay, that is how you can remember the presentation. Whereas in Angelman syndrome, it is going to be ataxia and seizures. Okay, remember the CNS pathologies will be with respect to mother, that is M is deleted. Okay, so mother's gene is deleted, meaning CNS. Okay, the father gene is deleted, then it is going to be obesity and hyperphagia. Okay, in that way, you remember, so it will be easier for you. Okay, then moving down, the disorders which are inherited. Okay, we discussed about disorders of gene imprinting, whereas we'll now discuss about inherited disorders. Okay, in inherited disorders, definitely would have known autosomal dominant, resistance, what is this, X-linked dominant, X-linked recessive and all. So let us see one by one. Okay, only the important stuff I want you to learn. Rest of the things, like there is too much memorable stuff here which is too much volatile. Probably at the last moment, if you want it, you learn. Okay, so in not many of the exams they have asked. In Neetpg, I guess they have never asked this examples of autosomal dominant, recessive and all. So you need not learn about it. Okay, the examples I'm telling you about. So just remember to like identify what type of pedigree chart they are asking about. Okay, I'll tell you. Like it is very kinda easy, but it depends upon you and your ability to identify it. Okay, so if all the generations are affected, then it is going to be dominant. Okay, so you think it in that way. Just whenever you see a pedigree chart, if all the generations are affected, then it is going to be dominant. Both in X-linked and in autosomal, it is going to be dominant. If there is skipping of the generations, one generation is there, other generation is not there, then it is going to be most probably recessive. Okay, in that way it is better, you could diagnose it. Okay, then autosomal dominant, here all are affected. That is what I was telling, right? So in it is not about male or female. All the sex will be affected. Males and females will be affected. Everyone will be affected basically in autosomal dominant. So one of the best example is achondroplasia. Remember achondroplasia, Peter Dinklage from GOT, right? I told you about this in orthopedics, right? So achondroplasia will be autosomal dominant, because remember in that way Tyrion Lannister was dominant, right? He was the most dominant fellow in the entire GOT, right? From the beginning of first episode till the last episode, Tyrion Lannister was the one who survived everything, right? So you remember in that way. So you will be sorted. So achondroplasia, autosomal dominant. Then when it comes to autosomal recessive, here there is skipping of generations. Okay, so one of the best example is cystic fibrosis. What is cystic fibrosis? So there is CFTR gene defect. Okay, remember this, the gene CFTR gene defect. Okay, only the genes I tell you to remember. Please remember it, and if you want it, make a sticky note for all the genes that I tell you to remember. One is, I told you, right, Marfan syndrome was filigree gene. Okay, filigree gene, so that you remember. And then when it comes to cystic fibrosis, it is CFTR gene defect is there. And one more thing I want you guys to remember is that ATP 7A and ATP 7B gene. Okay, please remember that also. And in osteogenesis imperfecta, there is COLA 1 and COLA 2 defect that was there, right? So remember that thing also. Okay, so coming back to cystic fibrosis, it is going to be CFTR gene that is affected, where there is chlorine channel that is being affected in the body. Okay, so chlorine channel is very important in sweat glands and in GIT. Okay, so here what happens is, In sweat gland, the chlorine channel was like supposed to go inside, and it was supposed to be suppressed. Okay, whereas in GIT, the chlorine channel should be increased and there should be increased secretion. Okay, but what happens if there is channel defect? What happens is, in sweat there is gonna be more chlorine, and in GIT there will be less chlorine secretion. So what happens is, so there will be increased sodium chloride secretion in sweat, whereas in GIT the secretion of the chlorine has been decreased. So the other secretions will also be decreased, which will cause constipation because there is no fluid secretion in GIT. So this is the main pathognomic of cystic fibrosis. Okay, remember this. In sweat there will be increased chlorine secretion, that is why there is more sodium chloride and salt secretion. Okay, whereas in GIT there is lowered secretion, which will lead to constipation. Okay, that's how you remember with respect to cystic fibrosis. Then coming down to X-linked dominant, see again, dominant meaning all the generations will be affected. That is how you identify. If X-linked is there, then just look for whether mother is passing to everyone, or what the father is doing. Okay, so if a father is carrier, then 100% the daughter will be affected. Okay, then if the father is carrier, and 100% of the 0% of the son will be affected. Okay, if the father is just a carrier, then 100% of the daughters will be affected, whereas 0% of the son will be affected. Whereas X linked, right, X will be dominant, so from mother only will get the X, right? So X-linked dominant, mother, all are affected. It doesn't matter son or daughter, both of them will be affected. Okay, so that is how you identify X-linked dominant. One of the best example is fragile X syndrome. Okay, trinucleotide CGG. Okay, so remember it as CGG gene. Okay, CGG gene defect, which is X-linked dominant, will be presenting with congenital gigantic gonad. Same name. Okay, CGG. Okay, gene defect. Okay, same presentation, congenital gigantic gonad, CGG. Okay, remember it that way, and it will be very easy for you. And one more clinical feature will be IQ will be very low in this patient. Low IQ patient with congenital gigantic gonad patient, it is fragile X syndrome, which is X-linked dominant. Okay, then coming to X-linked recessive, it is gonna be Duchenne muscular dystrophy. Where did we learn about dystrophin? Integral protein, right? We were discussing about this in general physiology. I was telling you, right, there are three types of protein: integral protein, the surface protein, and lipid anchoring protein, and all these things we discussed, right? Surface protein, the spectrin defect I told you I will discuss in RBC. The integral protein, the dystrophin, if it is defected, then it is gonna be Duchenne muscular dystrophy. What is this? Okay, where here the children, right, mostly it will be children who are affected here. Most of all these congenital disorders or the inherited disorders, from childhood itself the features will be seen. Okay, so in X-linked recessive Duchenne muscular dystrophy, when the dystrophin is affected, we can see the muscles that are affected. Okay, most common will be lower limb proximal muscles which are affected. Lower limb proximal muscles. What are the lower limb proximal muscles? The girdle muscles, the pelvic girdle muscles, and the thigh muscles, not the leg muscles. Okay, please remember this. Okay, so that is why there is difficulty in standing up. Standing up was supposed to be supported by this girdle and thigh muscles, right? So that is why there is they are having very difficulty in standing up. Okay, so that is why we can see something known as Gower's sign here with respect to Duchenne muscular dystrophy, where the calf muscles. Okay, they try to use calf muscles and stand on all four limbs. Okay, they'll use upper limb and calf muscles to stand up. That is why their calf muscles will be increased in size. Okay, their calf muscles, the gastrocnemius muscles and all, will be increased in size just by looking at the patient itself and the way they stand up, and they try to get up. No, you can easily identify which type of disorder this particular patient would be having. Okay, so Gower sign will be there. Then anyway, the girdle muscles are affected. We discussed in the upper limb, right? So Trendelenburg, the waddling gait, Trendelenburg gait. Okay, so waddling gait can be seen here. Okay, so that's it with respect to Duchenne muscular dystrophy. Need not learn, what my suggestion would be, need not learn every inherited disorder, just these four. Autosomal dominant, one is there, that is achondroplasia. Then autosomal recessive, I told you, right, cystic fibrosis. In that way, only one example you remember. If they ask example identification and all, in these in they'll give four option, and which of this is autosomal dominant. It is all memory based questions. Okay, so they won't be asking any memory based questions as per my knowledge with respect to Neetpg. Okay, now as we are turning to clinical based questions, it is gonna be more of this clinical oriented rather than memory based testing. Okay, so that's it with respect to biochemistry of all these things. Okay, we discussed about nuclear biochemistry, you got to know all the stuff, and then we went on to discuss about the cellular biochemistry, then we discussed about genetics. Okay, these three topics are done with respect to biochemistry. Now let us move on to metabolism of biochemistry, the major thing that we all are worried, right? In this one single chart itself, I'll clear off your entire biochemistry and all the disorders that is presenting to you with respect to metabolism of biochemistry. Okay, we'll be doing that now. Okay, so this is how we are gonna approach the metabolism biochemistry. Okay, so the food we eat will be having macronutrients and micronutrients, right? So macronutrients will be carbohydrates, proteins, and fats. Micronutrients will be vitamins and minerals. Okay, so we'll start with macronutrients and then we'll go to micronutrients. So what happens just after we consume a food? Suppose we are eating a nutritious food which contains everything, that is carbohydrates, proteins, fats, and all the micronutrients. So let us see one by one how the body will process and what happens to these macronutrients and micronutrients. And also we'll discuss about the deficiencies and disorders when this will be affected. Okay, so when a pathway is affected, let us see what are the disorders that will occur. So starting with carbohydrates, suppose I'm eating food and carbohydrates. Let us take the best example of rice. Okay, so rice I have eaten, and what happens is, so when it reaches the intestine, we'll discuss what happens with respect to intestine absorption and all when we are discussing about the GI physiology. But for now, what we'll discuss is when we get carbohydrates, suddenly the insulin secretion starts happening. Okay, from pancreas, you know that, right? So insulin secretion starts happening. Then and there, a process called glycolysis will start happening. Okay, so what happens is all the glucose that is absorbed is being taken to liver and all the muscle tissue and all these things. Okay, before that, we'll be discussing, before we discuss about muscles and all, so let me tell you, all these pathways and all is happening in liver. So all the things that I'm discussing is happening in liver, think it in that way. Okay, so liver is where all these pathways will be happening. So now insulin has secreted and insulin will start a process called glycolysis. Okay, glycolysis is nothing but breaking the glucose into energy. Okay, so why do we eat food? We need energy, right? So that is why we eat food. Okay, so glucose, what it's happening is it is undergoing a process called glycolysis to produce something called ATP. Okay, ATP is the currency. Why do we go for work? For money, right? In the same way, why does someone has to go to glycolysis? Is to produce money, ATP. Okay, that's it. That is the end goal. Okay, so let us see what all happens. So glucose is there. So glucose will undergo glycolysis with the help of glucokinase and hexokinase. Okay. So in this hexokinase will be in most of the tissues. I told you, right? When we are talking about liver, so that is why glycokinase is important. Whereas elsewhere, it is the hexokinase that is important with respect to glycolysis. Glycolysis won't happen only in liver, okay? It will be happening elsewhere in other tissues also. But we are more focused with respect to liver, okay? This is very, very important. Please remember these two enzymes, that is glucokinase and hexokinase. Glucokinase will be happening in liver, okay? So glucose I have eaten and it has gone to liver. Now glucokinase will break down the glucose into glucose 6 phosphate, and then entire glycolysis pathway is there. You need not remember the entire glycolysis pathway. Okay, please remember this. You need not remember everything. Okay, now glucose 6 phosphate it has converted into. Now it will go into glycolysis process through an enzyme called phosphoglycerate kinase. Only the enzymes I tell you to remember, you please note it down, okay? Else any other thing you need not remember. Phosphoglycerate kinase, okay? See, this entire this entire chart I am giving it to you only, right? So note down only the all and everything that you need not take down, okay? Only the things that I tell you to remember is what you will have to note down, okay? Don't waste time, okay? Phosphoglycerate kinase is very important enzyme. I'll come back to you why it is, okay? So remember, now the glucose I have written, it is converted into glucose 6p by these two enzymes, glucokinase in liver, whereas hexokinase in most of the tissues. So glucose 6 phosphate has happened, then it is converting into pyruvate through phosphoglycerate kinase pathway, okay? Phosphoglycerate kinase is the enzyme, so which will further down many other things will happen. So finally pyruvate is there, okay? So what the pyruvate does is it will form something known as acetyl CoA. Please remember this, acetyl CoA, the most important enzyme, okay? Acetyl CoA, the most important thing that will be forming in the entire body, okay? Acetyl CoA. Then what happens is it enters the TCA cycle. You know TCA cycle, right? Krebs cycle, the most important. You'll see in all these Instagram reels and all. So all these years we have been learning since our 11th, 12th, etc. But we have never understood what is Krebs cycle, right? So I'll make it easy for you. Nothing but acetyl CoA is finally formed. It wants to form energy. So this energy formation is mainly through TCA cycle. What is it? So acetyl CoA is converted to citrate and it will enter the TCA cycle and produces energy by going into electron transport chain, okay? So finally 32 ATPs will be formed, okay? So this is the entire process. Glucose is converted to glucose 6 phosphate, then to pyruvate. Pyruvate will form acetyl CoA. Acetyl CoA, if there is PDH, wait, we'll come back to PDS later. So pyruvate will convert to acetyl CoA. Acetyl CoA will enter the TCA cycle through citrate. Like citrate and acetyl CoA combined together, then it will form. It will go into TCA cycle, and then finally it will go to some one more cycle called ETC, the electron transport chain, where finally 32 ATPs, one single carbohydrate molecule will form 32 ATPs. Okay, so one single carbohydrate molecule will form 32 ATPs is very much important. Okay, this you remember. Okay, this information about 32 ATPs is important. Suppose, let us see, this is the normal pathway when there is no energy in our body, we need energy. This is how the process takes place. Now suppose glucose is there. What else can the glucose undergo into other than producing energy? Let us see one by one. Okay. Suppose glucose is there, it can undergo HMP shunt pathway and form ribose 5 phosphate. Ribose 5 phosphate is important for DNA, right? DNA formation. DNA formation, if there is ribose 5 phosphate only, the DNA is deoxyribonucleotide, right? DNA. So that is why ribose 5 phosphate is very much required. So this pathway that the glucose follows is called HMP shunt pathway. Okay. So this is where glucose would go into, other than going into formation of ATP. Then, if not, suppose the glucose 6 phosphate would form, there is too much glucose. Okay, so too much glucose is there and too much energy is also not required. So that is when many of the glucose molecule will go and form glycogen in liver. Okay, in liver, this is the storage form of the glucose, storage form of the glucose. Okay, the reason why it is stored in the glycogen form, because it is complex. Okay, it can tightly bind and remain very tightly in the liver. Okay, so whenever it required, it will convert into glucose again and will be utilized for energy. Okay, so that is why glycogen form. Remember this, the reason why it is stored in the glycogen form is because it is in the complex form. Okay, so that's it. So glucose can undergo formation of ribose 5 phosphate, it can undergo formation of glycogen, and suppose if PDH is there, phosphate dehydrogenase, then and if it is anaerobic, suppose there is no enough oxygen for glycolysis, then it will go the formation of lactate. Okay, when exercise and all these things, right? So during exercise, not much of oxygen would be available sometimes. So that is when the lactate through lactate formation, the energy or the ATP will be produced. So this is one more pathway. If suppose it is an anaerobic environment that is present for the cell, so that is where lactate formation will happen. Okay, then suppose now acetyl CoA is formed, and if enough ATP is there, so suppose the body tells there is enough ATP for me, no need of further more ATP that you need to produce. So what happens is, then this particular glucose has now already formed citrate, right? So it will undergo fatty acid synthesis. Okay, if there is enough ATP, then it will undergo fatty acid synthesis. Okay, so what happens is acetyl CoA is there, then the citrate shuttle. I'll come back to you for with respect to how the fatty acid synthesis will happen, but for now you remember there will be fatty acid synthesis. Okay, then I was telling you about phosphoglycerate kinase, right? This enzyme is very important because glucose 6 phosphate can form glycogen, and from glycogen back to glucose through this enzyme. Okay, this is the enzyme that is forming the two things, that is glycogen formation is also through this enzyme only, and if you want to break down glycogen and form glucose also, this enzyme only is important. So that is why phosphoglycerate kinase is very important. So now that we have learnt that the ATP formation is happening through glucose. So let us see, if there is enough ATP, I told you, right, there is fatty acid synthesis. So let us see how this fatty acid synthesis happens. Okay. So now that the acetyl CoA is there, okay, so what happens is citrate is there. So citrate will form acetyl CoA. Okay. So how will this acetyl CoA travel outside? Look, this fatty acid synthesis should take place outside. Okay, so outside the mitochondria. So mitochondria is there. Outside the mitochondria only this synthesis should take place. Okay, so how will this happen? So there is something called citrate shuttle. Okay, so citrate shuttle is there, which will carry this acetyl-CoA outside to the cytoplasm. This is the cytoplasm. So to cytoplasm, this citrate shuttle is what carries this acetyl-CoA. Where was this acetyl-CoA from? See, glucose from pyruvate had formed acetyl-CoA. Now the enough energy is there. Cell is telling enough energy is there. Mitochondria, I don't need enough too much money is there. So no need. Whenever I ask you, you please give me. I have glycogen also. You please store it in any other form. That is black money. Okay. So this black money is fat. Okay. So that is how the fatty acid synthesis happens. Okay. So this black money storage is nothing but fatty acid synthesis. Okay. So now for black money to form, what it has to do is through citrate shuttle this has come out. Now the acetyl-CoA, it will convert back to acetyl-CoA. Then this acetyl-CoA through acetyl-CoA carboxylase will form malonyl-CoA. Okay, malonyl-CoA will form a fatty acid, that is 16 carbon chain fatty acid, that is palmitate. This is very important. Palmitate, 16 chain fatty acid is what the storage form or the most probable fatty acid that the glucose will form. Okay, fatty acid synthesis is finally the palmitate. Okay, that's it. It will either store in the liver or pack as triglyceride in VLDL. Okay, so we'll come back to this, what is this VLDL and all, but for now you remember, now the fatty acid synthesis has happened in the liver outside the mitochondria, that is cytoplasm, and now it has to store. It will either store in the liver or it is going to pack as triglyceride and will be moved out of the liver through VLDL. Okay, so we'll come back to that VLDL later. So for now, so now that we know how the fatty acid formation is there, so let us move on to some other stuff, and then we'll come back to how the fatty acid breakdown will happen after learning about how the fatty acid that we intake through the food will also come in. Okay, so we'll discuss about the fatty acid thing later. So let us move on to a different topic of proteins. Okay, so in proteins, it is nothing but after the breakdown through enzymes and all we'll discuss in gastrointestinal tract. Okay, so when we are discussing about gastroenterology, I'll be telling you what are the enzymes that are important in breakdown of the proteins. Okay, so the finally it's gonna be the amino acids that are important. What are these important amino acids? Okay, so let us discuss one by one. Okay, amino acids are of two types. Okay, one is essential amino acids, which are taken through the food, because these amino acids cannot be generated in the body, so that is why it should be given through the food. Okay, so these are essential amino acids, whereas the other non-essential amino acids will be already there in the body and they can be utilized as such. Okay, so what are the essential amino acids? That is remembered through Private Tim Hall. Okay, so this is the like mnemonic that is required for essential amino acid. You need not remember what are all those things. Okay, so this is not a memory test. Okay, so they are moving on from memory test to clinical based questions, so that is why it is not much of an importance. If you want it, you can learn it just by opening your protein metabolism chapter from any of the biochemistry book. First thing they will give you is Private Tim Hall, that is essential amino acids. So let us discuss what are the amino acids that are very much of importance. Okay, so one of the major thing is phenylalanine, because this produces many other amino acids which are important for various other stuff in our body. So phenylalanine will form tyrosine. Okay, so and from tyrosine dopa will be formed. Through dopa melanin will be formed and dopamine will be formed. Through dopamine, norepinephrine and epinephrine will be formed. Okay, remember melanin, we had discussed it in dermatology with respect to tyrosine. Tyrosine deficiency will cause albinism. This is what I was telling you about, because see, tyrosine is what is required for melanin formation. If there is tyrosine deficiency, there occurs a disorder called albinism. Okay, that's it. Okay, phenylalanine will form tyrosine, dopa, dopamine, norepinephrine, epinephrine. All these catecholamines are very important, right, for our body. So that is why phenylalanine is very much important. Okay, suppose there is increased formation of phenylalanine because it is not getting converted to tyrosine. What happens is phenylketonuria, PKU. This you would have learned hundreds of times in your first year, right, for your theory exams. And also PKU, phenylketonuria, is the most important stuff. Okay, there are two types of phenylketonuria: classical and non-classical. Okay, in classical, it is going to be the phenylalanine hydroxylase enzyme deficiency, whereas in non-classical, it is going to be BH4 or dihydrobiopterin, which is the enzyme that is defect in non-classical type of phenylketonuria. Then suppose in tyrosine, see, tyrosine, too much tyrosine formation will be there. Okay, so most of it will go to the formation of dopamine, epinephrine and norepinephrine, but some of it has to degrade, right, because it is not much important and it should degrade. Okay, suppose if there is not enough degradation, or the degradation pathway is affected, then it will lead to something called as AKU, alkoptinuria. Okay, remember this alkoptinuria. Please do not confuse this with phenylalanine ketonuria. Okay, PKU and AKU are different. Please do not confuse these two things. If tyrosine degradation is affected, then it is gonna go into AKU formation, whereas phenylalanine degradation into tyrosine is not happening, then it is going to be PKU. PKU has two types. Please remember these two enzymes. Okay, BH4 and phenylalanine hydroxylase. Okay, that's it. So we were discussing about alkoptinuria, right? So alkoptinuria is gonna present with black urine. This is the clinical feature that they'll give you. Then black sclera. Okay, a child comes with the long-standing black urine and all this presentation. Okay, close your eyes, go for AKU. Okay, that's it with respect to phenylalanine. Then the other amino acid is tryptophan. Tryptophan will give rise to serotonin, melatonin and niacin. Tryptophan will form serotonin. Serotonin will form melatonin. What is this melatonin is important in sleep cycle, right? Circadian rhythm. Melatonin is important in sleep cycle, whereas niacin is important in NADP formation, the energy cycle. Okay, suppose if there is increased serotonin in our body, it will lead to a syndrome called carcinoid syndrome. Okay, carcinoid syndrome. Please remember, it is because of increased serotonin formation, because of tryptophan or some other things also can increase the serotonin and form carcinoid syndrome. Please, just by the name, you do not think that it is some disease of cancer or something like that. Carcinoid syndrome is the name given for increased serotonin, okay, in the body. Then coming down, it is gonna be glycine, which will form heme. Okay, glycine and succinyl CoA together will form heme. Heme formation we'll discuss in other chapters, like for example we'll discuss in hematology basically during the formation of RBC and heme. But for now, remember, glycine is also an amino acid which will combine with succinyl CoA to form the heme. Okay, then arginine. Definitely would have seen this question in any of your question bank. Arginine, nitric oxide. Nitric oxide is formed by which amino acid? Arginine. Okay, arginine will form nitric oxide, creatine and urea. Then coming to glutamate, glutamate will form GABA. Glutamate is an excitatory neurotransmitter. GABA is a inhibitory neurotransmitter. Glutamate is what will increase the neurotransmission. For example, if I want to move, glutamate will brain will release glutamate. All the neurons will release glutamate. Okay, whereas if I want to inhibit some movement, it is going to be the GABA, which is the brain will release or the spinal cord will release. Okay, so spinal cord will release glycine. Okay, not GABA. It is gonna be glycine which spinal cord will release. It is not GABA. Inhibitory secretion or the inhibitory neurotransmitter of spinal cord is glycine, whereas elsewhere in the body it is going to be GABA. Okay, remember this. These are the amino acids that the that are from the proteins. Okay, so that we ate through our food. Okay, so that is why amino acid is important. It is the building block. All the enzymes, hormones, all the proteins in our all the building stuff, everything will be from the proteins that we eat. That is why Indians are having protein energy malnutrition, right? Because all these enzymes are defect and all these metabolisms, everything will be affected, right? So that is why it is very important to eat not only carbs, because I told you, right, enough carbs, when enough carbs are there, so black money storage will happen. Okay, so that black money storage is fatty acid. Fatty acid is nothing but the adipose tissue, the tire around our belly, that will be there, right? So all these things, or the visceral fat storage, everything will happen because if we start eating more carbohydrates. Whereas if we start eating more protein, there is not much of a metabolic disorders that will happen, right? So this is a topic for discussion for some other day, but for now you remember amino acids and the proteins are very much important for our body. Then coming to urea cycle. What is urea cycle? Now all these amino acids and proteins that we talked about should be broken down, right? Should be broken down and should be thrown out of the body, because suppose if all this starts storing inside our body, it is going to be toxic to our body, right? So that is why it should be broken down and thrown out of the body. What is the throwing out substance after the formation of, I mean after the utilization of the amino acid, is NH3 or ammonia. Ammonia is the final form of all these amino acids when they start breaking down. Okay, so ammonia is what we throw out of our body, but ammonia itself is a toxin. It cannot be stored inside our body as such, or cannot be thrown outside of the body as such. So that is why this ammonia toxin material should be converted into intoxic material called urea, okay, before throwing out. That is why our body has created a cycle called urea cycle, where the detoxifying organ, which is the liver detox. Remember, detoxifying organ of our body is liver. So all this ammonia will travel to urea, travel to liver, where this will get converted to urea and it will be thrown out through kidney and urination. Okay, so this is how the entire process of urea cycle is there. So this is the basics of a urea cycle, the skeleton of the urea cycle that I told you about. So let us go deep into it. It is very easy. Urea cycle has been an headache since our first year, but let me make it easy for you. Okay, so I just now I told you the skeleton of the urea cycle. So let us now dive deep down. Okay, now amino acid has broken down, protein formation is there. Okay, so I mean protein has broken down, so now it has to undergo amino acid formation. So these proteins that are broken down are converted to ammonia in that particular cell. For example, let us take brain, let us take muscle tissue. All this protein breakdown will form ammonia. Okay, so now I told you, right, ammonia is toxic. It should not stay even for a minute over in that particular cell. So immediately what happens is there occurs a cycle called Cahill cycle, where this NH3 is converted to carryable form. What are these carryable form? It is glutamine and alanine. Glutamine is the carryable form of ammonia in brain. Okay, please remember this. Whereas alanine is the carryable form in all the other parts of our body. Okay, now after converting into carryable form, it will go down to liver. Liver, I told you, right? This is the detox organ. Now that it has reached liver, it is safe that it can convert back to ammonia, because here also it is not gonna keep the ammonia as such, it is immediately converting it to urea. Okay, so how is this ammonia conversion will take place? Let us see. So ammonia will combine with carbon dioxide and bicarbonate and form something called as CPT1. Okay, CPT1. Okay, so through CPT1, this carbamyl phosphate synthesis will happen. Carbamyl phosphate with OTC will form ornithine. Ornithine will form aspartate, and aspartate will give one more ammonial or one more nitrogen molecule and will be converted into arginine. Arginine, I told you, right, will convert to urea. We were discussing about arginine, right? Nitric oxide, urea, and all these things, right? So arginine will be converted to urea, and in urine, this urea will be excreted out. So you got it, right? Broken down ammonia is dangerous, so it was converted to carryable forms of glutamine and alanine in that respective cells. It has come down to liver. Here the detoxification is happening, the urea cycle basically. Urea cycle is nothing but ammonia is combining with the carbon dioxide and bicarbonate together with the CPT1 and OTC. They'll go and form ornithine, and the urea cycle will take place. Finally arginine will form urea, and urea is excreted through urine. Okay, this is the entire urea cycle. Suppose if there is a defect in CPT1, CPT1 we saw it here, right? Carbamyl phosphate synthetase activity, right? So in the urea cycle, if CPT1 deficiency is there, then ammonia can't be converted into urea. Ammonia starts accumulating in liver. No, in liver it has already reached. Okay, in liver this enzyme deficiency is there. Ammonia starts accumulating in liver, so it is gonna be dangerous, right? Ammonia is dangerous, so it is gonna cause hyperammonemia type 1 if CPT1 is deficient. If OTC is deficient, then it is gonna form hyperammonemia type 2. That's it. This is the major thing that you wanna learn about. In hyperammonemia, what is the treatment is lactulose and phenylbutyrate. What is the mechanism of action is very much important. Okay, lactulose and phenylbutyrate. Lactulose, what it does is it goes to gastrointestinal tract, and in gastrointestinal tract it will start removing the ammonia. Okay, so the ammonia that is formed by all this food digestion and all the damages that the food or some materials will be doing, all this ammonia, so it is basically acting like a potash alum. Potash alum, like basically it removes all the dirt material and all, right? So in the filtration, so in the same way lactulose is pulling down all the ammonia that is present in the gastrointestinal tract. So that is when the demand for ammonia will increase for lactulose. So that is when it will throw out all the ammonia that is present in the body. Then when it comes to phenylbutyrate causes increased like it combines with glutamine in brain, and what it does is, so glutamine requires ammonia, so it increases the demand of ammonia. Okay, so too much ammonia is required for phenylbutyrate and with glutamine. So that is when the ammonia excretion also start happening with phenylbutyrate. Okay, so these are the two mechanisms of action with respect to hyperammonemia treatment. Okay, that's it. So this is all and everything you need to know with respect to protein. So you got to know, right? Proteins, many amino acids were there, and what are they forming into? Then amino acid breaking down is happening. Then urea cycle happened, and urea was thrown out of the body. That's it with respect to protein metabolism. In carbohydrate metabolism, we discussed, right? Glycolysis, when energy was required. When energy was not required, it went into other formations. That is DNA formation, ribose 5-phosphate formation. Then it went on to form glycogen, because too much glucose is there. I'll store some of it as glycogen. The body tells, and when there is too much money too much ATP is there, then it was black money formation. That is fatty acid synthesis. Okay, that's it with respect to carbohydrate metabolism. Now let us move on to fatty acid metabolism. So now I've eaten fat. So what happens? Let us see. Okay. So the fat reaches the intestine. So once it reaches duodenum, bile acids start secreting. Okay. So bile acid and how it starts secreting and all we'll discuss when we go to gastroenterology. As I told you with respect to carbohydrates and proteins, everything enzyme related we'll discuss in gastroenterology. But for now, so let us take the bile acid already started excreting, and now the chylomicron formation is has started. Okay, chylomicron formation has started. So this chylomicron will form something known as this chylomicron combines with HDL. Okay, so there is something called HDL. Okay, high density lipoprotein. Okay, this HDL gives something known as ApoB48. Okay, ApoB48. This HDL gives ApoB48 to chylomicron. Finally, this chylomicron will look something like this. Okay, so what is this? So chylomicron will be having a ApoC2 and ApoE. Okay, all the chylomicrons will be having ApoC2 and ApoE. Okay, HDL gave one more apolipoprotein called ApoB48. B48. Okay, so after formation of this entire chylomicron structure, this will go to liver. So from enterocyte circulation, it is going into liver, right? So here in liver, there is something called as lipoprotein lipase, LPL. Please remember this, LPL. So here in lipoprotein lipase, what it does is it will recognize the ApoC2 that was present. I told you, right? There are three types of receptors in the chylomicron. Okay, ApoC2, ApoE, and ApoB48. ApoB48 was given by HDL. What happens is lipoprotein lipase will recognize this ApoC2 and takes it inside. Okay, takes it inside the cell. Here, this entire chylomicron will be broken down into triglyceride and fatty acid. Okay, some of them will be triglyceride and some of them will become fatty acid. Okay, the remnants of this broken down thing will have ApoE and ApoB48, because ApoC2 it recognized and all the chylomicron has broken down into triglyceride and fatty acid. The remaining thing was there, the remnant was there, right? So that will be recognized by the liver receptors by ApoE site. Okay, so ApoE and ApoB48 has been remained because ApoC2, right? ApoC2 was recognized by lipoprotein lipase and it was taken down. So here is where the liver will recognize ApoE receptors and start taking up. Okay, so finally all the triglyceride, chylomicron, all the fatty acid that was consumed by me is taken down by liver. Okay, so taken up. Now I told you, right, here there was formation of triglyceride and also the formation of fatty acid. So what does it happen? Look, what finally happens to that triglyceride and fatty acid? So the fatty acid that was formed will be stored to utilize later. Okay, just like fatty acid synthesis that we discussed when carbohydrate was increased, right? Like we discussed, right? Carbohydrate will the black money thing, so the same thing, it will store and will be stored for utilization for the later part. Okay, so let us discuss what happens to the triglyceride. So this triglyceride, so whenever required by the body, will form VLDL. We discussed this also, right? So here in carbohydrate, we were discussing, right? One thing it happens is storage form. The fatty acid was stored. The other one is pack as triglyceride in VLDL. So this is the exact thing I was talking about. This happens, the same fatty acid thing happens in carbohydrate also if there is excess carbohydrate, and also when we eat fat also the same thing happens. So that is why carbohydrates and fat are not much different things. Those are two same things. Okay, carbohydrate is a carbon chain molecule, right? This is also a carbon chain molecule. So entirely same things. Okay, just a little bit here and there, but finally it is gonna go and form the adipocytes only. Okay, so we are discussing, right? We'll move on. Okay, so VLDL formation is happening. So what does VLDL formation further go into? So remember, HDL gave ApoB48 to the consumed chylomicron, whereas here the VLDL formation is by liver. So here HDL gives ApoB100, okay, to VLDL. To chylomicron it gave B48, whereas to VLDL it is giving ApoB100. Okay, this is the ApoB100. So finally the VLDL structure looks something like this. ApoC2 is there, ApoE is there, and ApoB100 is there. Finally, what happens is the liver starts secreting VLDL whenever required. So whenever required, what does that mean? So it is nothing much. Like liver can't store much of the fat. Okay, so it has other duties also. Urea cycle is there, other many duties are there. It has to produce too many hormones. Everything is there. Everything it can't do, right? Storage of fat also that can't do. So that is why in our body what it does is you go and store somewhere else. Okay, so what it does is it packs as VLDL and goes and stores somewhere else, not in liver, but it will tell go and store in somewhere else. For some particular people it will be around the belly, some particular people it will be in the gluteal region, some people it will be in the face, and some people it will be in the breast. Okay, so this is how the division of it depends on different persons. Okay, so some people will be having too much belly fat, whereas some people will be having too much visceral fat in that way. Okay, this is the VLDL. So that is why they tell you, right, VLDL and LDL. I'll tell you what is LDL. So VLDL and LDL are danger cholesterols, whereas HDL is good cholesterol. Okay, so this is where it comes in. Okay, so now that it has packed into VLDL, it is gonna convert into adipocyte. Okay, how is this how is this happening? Same thing, just like LPL had recognized ApoC2, right? Here also adipocytes will recognize ApoC2 and it will take it in. So finally, the triglyceride starts storing as adipocyte. Then what happens is the same thing, remnant, ApoB100. There it was ApoB48. Here the remnant will be having ApoB100 and ApoE. So this remnant will be converted to IDL. So remnant, there it was liver that took up, right? So here a remnant, the liver took up. Here also same thing. Remnant is there. The remnant will be taken up by liver. Liver, what it does is after formation of IDL in liver, IDL formation will happen. This IDL will be converted into LDL and ApoE. Okay, ApoE will be broken down in liver, whereas LDL will remain in blood. Okay, remain in blood and in the peripheral tissue. That is why LDL is nothing but fat, right? It is cholesterol. Okay, cholesterol is bad, right? So the remnant of all this fat that was there, the amount of it, see, whatever was needed was stored, and some of it got utilized. Rest of it, what does it do? So what happens is it remains in blood. Okay, so that is why when there is increased of fat, that is increased fat, that particularly LDL. So that is why we see when cholesterol lipid profile we do, all the levels we see, right? LDL, HDL, VLDL. When there is LDL and VLDL level above certain point, that is when we tell that is it is dangerous, right? Not HDL level, we don't take the consideration of it. So that is why we tell LDL and VLDL, when that is increased in blood, it tells us that the peripheral tissues or adipocytes is not taking, there is too much fat in the blood. Okay, so that is when the LDL starts remaining in blood, right? When there is too much fat. Okay, so this is the entire metabolism of fatty acids. Okay, so now let us see what happens when there is fasting. This is all and everything that I discussed you when I am eating, right? So I ate glucose, it was converted to energy. When more glucose was there, fatty acid synthesis took place. When I ate essential proteins or amino acids, that underwent that particular enzyme formation or hormone formation, and remaining or the broken down stuff was going into urea cycle. Then I ate fat, where it went into formation or storage of fat. Certain of them remained in blood. All these things happened. This is when I was eating food. Suppose I'm undergoing fasting, I'm not eating food. So that is when the utilization of this stored fat, the fat storage we discussed about, right? Here in the liver, I told you, right? Fat was stored. Here also there was stored fat which was required for later utilization. Here also all these things, peripheral tissues and remained in blood, all these things was there, right? All these things, when there is fasting, these are the things that get utilized, because body is not getting energy from carbohydrates or fats, it has to get the energy from somewhere else. So that is when it will start utilizing the black money. Okay, black money is fat stored. Okay, so let us see what happens during fasting. So during fasting, in the first 1 to 3 days, first 1 to 72 hours, it is gonna be the lipolysis. Lipolysis, which will be the major thing that produces the energy. Okay, so lipolysis will give the energy, whereas more than 3 days, it is gonna be the ketogenesis. Okay, that provides the energy. We'll come back to ketogenesis later. For now, we'll discuss about the other forms of energy that we get during our fasting. So 1 to 3 days, it is going to be lipolysis and gluconeogenesis. Okay, so let us see what this gluconeogenesis is. Okay, so when we are not having glucose consumed by ourselves, so new glucose molecules will be formed either from glycogen or fatty acids or from proteins. Okay, so this is called gluconeogenesis. Gluconeogenesis occurs through fat. Okay, so there was stored fat, right? So here we discussed there was a stored fatty acid. So what happens is this stored fatty acids will be converted to free fatty acids and glycerol. What converts this triglyceride that was stored in our liver into fatty acids, free fatty acids and glycerol is hormone sensitive lipase. Okay, remember hormone sensitive lipase. When suddenly you are undergoing some 16-hour 18-hour fasting, someone told you one day a meal and all. So what happens is our liver starts secreting something known as hormone sensitive lipase. What it does is hormone sensitive lipase goes into the liver, tells brother, I need black money, because there is no money, the original money that I was earning is not there, so I need the black money that I stored with you. So that is when it starts releasing free fatty acids and glycerol. Okay, so now what happens is this glycerol will undergo formation of glucose, and the free fatty acids will start undergoing the other process and go to the liver. Okay, so let us discuss about glucogenesis first, and then we'll go on to lipolysis. Okay, or the fatty acid utilization, that is beta oxidation. Okay, so we'll discuss that later. Now let us discuss gluconeogenesis. The glycerol, I told you, right, storage triglyceride will be free fatty acids and glycerol. So this free fatty acids, the glycerol will be the will go undergo gluconeogenesis, where only the odd chain fatty acids are the only thing that can form glucose molecules. Okay, this is not the even chain. Remember this, please remember this. This is the odd chain fatty acids, the glycerol, are the only things that can form glucose. Okay, then now I told you, right, fatty acids, free fatty acids was there. These are the two molecules that happened from stored fat, right? So free fatty acids, then the other glycerol went into gluconeogenesis. For same thing, for gluconeogenesis, meaning glucose was formed, and later again, see, it is going back to formation of glucose, and glucose finally to 32 ATP. Okay, so gluconeogenesis was there. So then coming to free fatty acids, this also can produce energy. How can it produce energy? So free fatty acids from the liver or wherever it was stored will be carried by albumin to liver, heart and muscles. Okay, so after carrying this to liver, heart and muscle, what it happens is fatty acid co-synthetase will convert it into fatty acyl CoA. This fatty acyl CoA will go to liver or cytoplasm, and from there, to convert it into energy, it has to go inside mitochondria. Fatty acyl CoA cannot itself go into mitochondria. Mitochondria is the powerhouse of the cell. This, since you are first standard, you know mitochondria is the powerhouse of the cell. What does that mean? Mitochondria is the place where all this energy formation will take place. See, even in a TCA cycle, the 32 ATP, where did it happen? It happened in mitochondria, right? So, energy, meaning the production of ATP, production of currency, is happening in mitochondria. Okay, that is where the minting, RBI. Okay, so remember, mitochondria is RBI. Minting of all this money is happening in mitochondria. Okay, so to carry this fatty acyl CoA inside mitochondria, we need a shuttle. This shuttle is carnitine shuttle. Okay, remember, acetyl CoA, to move outside of the mitochondria, we needed citrate shuttle. Okay, do not confuse citrate shuttle with carnitine shuttle. Okay, citrate shuttle was required when acetyl CoA was formed and there was enough energy, and finally body decides, our liver decides that it should undergo fatty acid synthesis rather than more money formation. Black money should be stored. So that is when citrate shuttle came in. Here it is the carnitine shuttle. Here we need energy. There energy was not required. When energy was not required, that is when citrate shuttle came in. Whereas here in carnitine shuttle, here energy is required. That is why fatty acyl CoA is traveling to mitochondria. Here, after traveling inside mitochondria, this fatty acyl CoA degradation will happen. Okay. So, after degradation, it will form acetyl CoA. Acetyl CoA, the favorite substrate, right? Favorite substrate. What does it undergo into? Acetyl CoA is the main substrate that was required for TCA cycle, right? Finally, it will undergo TCA cycle. That's it. So, glucose was not there. We already had we already had stored our fat. Okay, we already had our black money stored. Now black money utilization should be done. How is it done? Either it should form the white money, or through other purchases we should convert that black money into main energy, main currency, right? So this is the two things that happens. One is glycerol was there, which formed the white money directly, whereas free fatty acid went on through all these things and finally formed the acetyl CoA to convert it into ATP. That's it. This is the fasting process that takes place when it is between 1 to 3 days fasting. Suppose I fast for more than 3 days. Okay, if some people are doing this fasting protest and all, so more than 3 days the energy should be required. Only till 3 days my stores will give the energy, but more than 3 days it has to undergo a process called ketogenesis. Okay, sometimes even I have done it. So there is something called as 72-hour fasting, right? So that is when we'll start the ketogenesis or ketone body synthesis in our body, so that all this stored fat or LDL, all these things should get mobilized, and that should go into utilization of energy, or else there will be too much storage of fat that is happening. Okay, so that is why we should always do fasting. Okay, I recommend fasting. Okay, so now let us see how this the fasting after 3 days. What happens after 3 days? Okay, so for pyruvate to form into acetyl CoA and to get utilized as energy, there we need something called as oxaloacetate. Okay, so oxaloacetate is one of the biggest and greatest substance that is required for all this metabolism. Okay, we were discussing more about acetyl CoA, we forgot about oxaloacetate. Oxaloacetate is one of the favorite and most important substrate that is required for all this TCA cycle to happen. When the oxaloacetate substrate, oxaloacetate, where did it come from? It is coming from glucose only. Okay, so now the glucose is not there inside the body, all the gluconeogenesis stuff also I have utilized. Where will the body get its oxaloacetate from? So nothing is there, right? So if oxaloacetate is not available, then the acetyl CoA formation is there, because there is too much fat that I have stored from all these years of eating. Acetyl CoA formation is there from the fatty acyl CoA just now we discussed, right? So acetyl CoA requires oxaloacetate to go into TCA cycle, but there is no oxaloacetate because there is no glucose at all. What will it undergo into? So our body has another mechanism called ketogenesis, where the acetyl CoA will deviate to ketogenesis through HMG CoA pathway. Okay, so acetyl CoA will be converted to HMG CoA through HMG CoA synthase. This HMG CoA will be converted into acetoacetate. Okay, this acetoacetate will be converted to beta hydroxybutyrate and acetone. Okay, so this is the pathway of ketogenesis. Acetyl CoA is there. Okay, it cannot undergo TCA cycle. Okay, so that is when it is converted into HMG CoA with the help of HMG CoA synthase. This enzyme is very important. Okay, so HMG CoA synthase is what converts into HMG CoA. Now this HMG CoA is converted to acetoacetate, and acetoacetate will be converted to beta hydroxybutyrate and acetone. Acetone will go to lungs. That is where we'll get the Kussmaul breathing, right? Remember acetone breath that we'll see in long-term fasting and all. So the investigation that we do for it is to test whether the acetone formation is there or not. So this is Rothra's test. See, remember Kussmaul's breath. Where can we say this? DKA, diabetic ketoacidosis. What happens is the body cannot utilize glucose because insulin is what was required, right? So first thing I told you was insulin, right? When I started talking about carbohydrates, insulin is what promotes all this glycolysis, etc. When the insulin is not there, on insulin resistance is there, it the body can't utilize glucose. It will store into fats and all these things. Okay, so body has to get its energy from somewhere. So what will it do? From if glucose is not there, oxaloacetate is not there, where will it get its energy from? Acetyl CoA will undergo ketogenesis. That is what the main pathogenesis behind diabetic ketoacidosis. Okay, so when we have a patient with diabetic ketoacidosis with glucose level in the blood as 500, because the blood was supposed to utilize, I mean, the body was supposed to utilize this amount of glucose, but glucose utilization is not there. So when more than this amount of glucose is present in the blood, it is definitely ketoacidosis, and we can see Kussmaul breathing. And also we can investigate through Rutra's test, where we can get the acetone positivity, okay, in DKA patients. Then I was discussing, right? So how will the body meet its energy requirement through ketogenesis? So beta hydroxybutyrate will travel to tissues, okay. So this will convert into acetyl CoA again in the after traveling to the tissues, and there it will undergo TCA cycle. In other tissues, TCA cycle can undergo, because there the oxaloacetate is there. Where did the oxaloacetate come from? So it had already stored, but in the liver it is not there. From liver only we need energy, right? So but from liver it is not there. So that is when it will have to travel to other tissues where the energy can be formed. So this is the travel form. Beta hydroxybutyrate is the travel form, where the acetoacetate will be converted and goes back to TCA cycle, and finally after TCA cycle 32 ATPs will be there. So this is how our body keeps ourselves active even during fasting. Okay. So till 3 days you won't die. Some people will be telling, right, oh, I can't fast for 5 hours, 6 hours. Oh, yo, morning I ate. I'm not. I didn't eat in the afternoon. I can't survive. It's gastric and this is all nonsense. Okay, so body can survive more with more fasting also, okay. And fasting is very, very, very good for health, because all this stored fat and all will be getting utilized through this method. This is the entire metabolism, biochemistry metabolism, that you had to learn. Since your first year, this has been an headache, right? So again, once again, please watch this video, and it will be very beautiful. If you want, make your own notes. This is my notes, and so this is the way that you have to learn all this metabolisms. So finally you got it right. I ate carbohydrates, proteins and fats. What was happening? What was happening in fasting? All these things you got an idea, right? So this is the entire process, how the food metabolism is happening. Okay, that's it. So this is with respect to macronutrients. So now that we are done with macro nutrient metabolism, let us see what are the micronutrients that are important, and what are the disorders that would happen when there is micronutrient deficiency. Minerals will discuss with respect to that particular systems and all. So only the minerals that are important are iron and copper. Okay, so iron, copper and zinc will discuss further when we go on to that particular chapters. Okay, so now let us discuss more about the micronutrient that is very important, that is vitamins. Okay, so vitamins are nothing but enzymes. Okay, so enzymes that are given a certain name called vitamins. Okay, so vitamin A deficiency, if it causes, it is going to be xerophthalmia or phrinoderma. Is a dermatological disorder where we'll be seeing in the extensor aspect some vesicles and all. Okay, so this is what we'll be seeing when there is vitamin A deficiency. Xerophthalmia, vitamin A, you would have definitely learned from your 5th standard also, that it gives eye vision, right? So all these things of bitoid spot, xerophthalmia and all these things you would have definitely learned in your final year. Just go through it once, and definitely some vitamin A deficiency is there, eye symptoms will be there. So that will be there in our clinical cases also. Okay, so that is how you can identify. Then when it comes to vitamin C, as I discussed in orthopedics, so collagen, right? Collagen formation, vitamin C is important. When collagen formation is defective, then scurvy scurvy, the bleeding gums, and all these things will be there. Okay, so it is also important in iron absorption from our enterocytes. When we eat iron, if we give vitamin C tablets, those people will be having better absorption. Okay, so this is the thing that I want you guys to keep in mind. When it comes to vitamin E is an antioxidant. Okay, when this vitamin E is deficient, then it is going to be acanthocytes. Okay, we'll discuss more about acanthocytes in hematology, but for now you keep in mind vitamin E is an antioxidant. When the vitamin E is deficient, RBC will be having instability. So that is why the breakdown of the cell membranes and all these things will happen. That is where the acanthocyte formation will happen. So that is why vitamin E is very important. Okay, to maintain antioxidant level in our body. Oxidants or the radical molecules will destroy our cells, right? So to make it stable, it is going to be the vitamin E that is very important. Then coming to vitamin K, it is epoxide. Remember, the word epoxide is nothing but vitamin K. Here the clotting factors 2, 7, 9 and 10. Okay, so the formation of 2, 7, 9 and 10 require vitamin K. Okay, so that is the thing I want you guys to remember. And A, D, E and K are fat soluble vitamins. Okay, so when there is deficiency of fats, or when there is difficulty in fat absorption, for example, let us take there is an obstruction in bile duct due to obstructed jaundice or something is happening. Leave about obstructive jaundice, I'm telling, the bile acid secretion was supposed to be there for fats, right? So fat was forming by fat to chylomicron was by bile, right? So if there is no bile secretion, how will the vitamin, that is A, D, E and K, how will they get absorbed, right? So absorption of ADEK cannot be there. So that is when the vitamin deficiency of this also can be seen when there is an obstructive jaundice. Okay, so this is one thing I want you guys to keep in mind. Okay, then when it comes to vitamin B, there are many different vitamins. Okay, so that is why the name vitamin B complex. We give B complex tablets to people with ulcers around the mouth, that is stomatitis, cheilitis and all these things, right? Why the term B complex is because we have many different vitamin Bs. Okay, so vitamin B is called thiamine, vitamin B2 is called riboflavin, and vitamin B3 is called niacin. Remember it as train. Okay, so vitamin 1, 2, 3, remember it as train, TRAIN. Okay, so train. Okay, so remember yes, it as train, then it will be very useful for you. What are they're talking about? Vitamin A, when thiamine is deficient, we'll be seeing beriberi, wet beriberi and dry beriberi. Wet beriberi is nothing but CVS abnormalities, whereas dry beriberi is CNS abnormality. Okay, remember in that way. Then there is Wernicke's encephalopathy. Then there is Korsakoff psychosis, ataxia, and all these things can be seen when it comes to vitamin B deficiency. Okay. Then when it comes to vitamin B2 deficiency, then it is going to be cheilitis and stomatitis. That is, I told you, right? B complex. Why do we give it? For stomatitis and cheilitis, right? So this is where we are targeting the vitamin B2 deficiency. Okay, then when it comes to niacin, your favorite thing, that is pellagra, dermatitis, diarrhea, death and dementia. Okay, so these are the 4 Ds of pellagra. Just see a picture of pellagra and you will be able to solve niacin. It is the niacin that they are talking about, that is train. Okay, then when it comes to B6, B7, B9 and B12, these all things almost look the same. Okay, B6 is required for many metabolism like heme synthesis, GABA synthesis and cysteine synthesis. Okay, so remember in that way, B6 PLP is required for heme, GABA and cysteine synthesis. B7 biotin, it happens because of increased egg white consumption in gym gym people. When people start eating too much white egg whites, right, they'll be having hair loss. We think that it is because of some testosterone or something he is taking, that is why hair loss is there. No, that is because of egg white. Okay, when there is increased consumption of egg whites, only egg whites, then this biotin deficiency will happen and there is increased hair loss. Okay, so biotin deficiency will be presenting with hair loss. Okay, then when it comes to B9, folate, remember folate, folic acid that we give in our pregnancy. So why do we give it? For DNA synthesis. B9 is very much important for DNA synthesis. If there is defective DNA synthesis, it might lead to megaloblastic anemia, because RBC has certain coding and all. When there is DNA synthesis issue, the RBC will form some random structure, right? So that is when the enlargement of the RBC can be seen. That is known as megaloblastic anemia, where we can detect that okay, there is DNA synthesis issue. Okay, so that is where B9 is important. Whereas B12, this is also same thing, DNA synthesis. It is helping in DNA synthesis with B9. Okay, so here also megaloblastic anemia we can be seen, and it is also helping in heme synthesis and methionine synthesis. One of the major things I forgot to tell you was, there is something called homocysteine. This is also an amino acid. Okay, homocysteine can be converted into cysteine and methionine. Okay, to convert into cysteine, it requires B6, whereas to convert into methionine, it requires B12. Okay, when there is no conversion, when there is a defect in any of the conversion, it will lead to something called as homocysteinuria. When we are detecting homocysteinuria in urine, we can see increased amount of homocysteine. So that is when you can tell that whether B6 or B12 deficiency is there. When B12 is deficiency is there, it is gonna affect the methionine formation. Whereas homocysteine issue is there, then it is gonna be cysteine formation issue. Okay, this is the major and everything that you need to know about macronutrients, micronutrients, its metabolism. That's it. Okay, guys. Okay, now coming down to the disorders that we can see in this metabolism is most of the metabolism disorders are discussed while discussing the metabolism itself. But let us see some specific metabolic disorders. One major thing is glycogen storage disorder. Okay, when so I told you, right, glucose was converting itself into glycogen because it is one of the storage form of glucose. Because when enough glucose is not there in the body, it might get its glucose directly from glycogen, right? So that is why it was the storage form of glucose. Now the glucose formation or the glucose storage has properly happened, but to convert back into glucose, there are some issues. So at each level of conversion of glycogen into glucose, if the enzyme deficiency is there, it is gonna cause certain particular clinical features. Okay, let us see one by one with respect to the enzyme that is defect. Glycogen is there, it is gonna form into glucose 1 phosphate, then to glucose 6 phosphate, then it is gonna convert into glucose. If in the first step if there is a defect, because glycogen to convert into glucose 1 phosphate, glycogen phosphorylase is required. When there is a defect in this, particularly myophosphorylase, then McArdle's or Hurst disease can be seen. Here it is gonna present with exercise cramps. Okay, exercise cramps. In the question itself, they will be telling a person with low glucose hypoglycemia, when he exercises, he is getting exercise cramps. So directly go for McArdle's disease. That's it. What is the enzyme disease defect? Particularly, it is myophosphorylase. In a previous year question, they had given glycogen phosphorylase and also myophosphorylase. Which will you choose from? Both are correct, but in particular, it is myophosphorylase that you have to choose. Okay, this is the question. This is the previous year question, and this exact thing was asked. Okay, all these features, everything was given. Even the McArdle's also it was mentioned, but they told us to choose from the enzyme that was defect. Okay, so this is the thing that you have to focus on now. Okay, the first step has passed. Glucose 1 phosphate has formed. Now to convert into glucose 6 phosphate, we require an enzyme called debranching enzyme. When debranching is a debranching enzyme is affected, so it is gonna be the Cory's disorder that we'll see here. The patient is going to present with hypoglycemia, mild hypoglycemia. Okay, so we'll be telling the baby will be lethargic, or in that way. When we measure the glucose, it will be like mildly low. Okay, so in that way it will be there. And when we do the metabolic disorder testing, it is gonna come back to debranching enzyme defect, and it will finally diagnose it as Cory's disease. Suppose there is a lysosomal disorder, and the debranching enzyme is affected. So this is where the Pompey's disease will come in, where the patient is going to present with cardiomegaly and floppy baby syndrome. Okay, so when there is these two particular clinical feature, please go for Pompey's disease. This is glycogen storage disorder. This is how you are supposed to study. Now, glucose 6 phosphate has formed. This is the third step that we are discussing about. So, to convert glucose 6 phosphate into glucose, we require glucose 6 phosphatase. When that particular enzyme is defect, then we will see something known as von Gehrk's disease, where the hypoglycemia was severe. Okay, so you would be definitely confusing between whether to choose von Gehrk's or Cory's disease. If it is a mild symptom, then it is gonna be Cory's disease. Where if it is a severe symptom, then it is gonna be hypoglycemia with respect to von Gehrk's disease. So that's it with respect to glycogen storage disorder. Then one more thing is there, that is Anderson disease, where there is a defect in formation of glycogen. Okay, so we discussed now is breakdown of glycogen. There are this issues is there. Okay, glycogen storage disorder, meaning there is abnormally increase in glycogen storage, right? Because glycogen is not able to get itself broken down. That is the disorder, glycogen storage disorder. Okay, but if there is a problem in formation of the glycogen itself, then it is going to be Anderson system, where when we test glycogen levels will be very low. Okay, so a patient will present to you with hypoglycemia, will test for glycogen, but the glycogen itself is very low. But here in all these other glycogen storage disorders, glycogen will be high, but we'll be getting hypoglycemia itself. Then when we test on further with respect to the enzyme that is defect, then we'll get to see these branching enzyme or debranching enzyme defects. Okay, so that is the difference between Anderson and all these other glycogen storage disorders. So then moving on further, we'll discuss about lysosomal storage disorders. Okay, what are these lysosomal storage disorders? There are many substrates inside and outside the cell, large and small, which are broken down by lysosome. When the enzyme required to break down these substrates is defected, it is called lysosomal storage disorder. So what does that mean? Suppose many substrates are there in our body which are supposed to be broken down by lysosome. Lysosome is like breaker of all these things. Okay, so all the substrates and all will be digested by lysosome. Okay, it is like a dustbin. Okay, so finally a vacuum which can clear out all these substrates. Okay, when there is a defect in it, it causes lysosomal storage disorder. So let us see what are all the lysosomal storage disorder and how it presents. Okay, Niemann-Pick disease. What does that mean? So there is sphingomyelin accumulation. Okay, sphingomyelin was supposed to be broken down. Okay, this is a substrate. If it starts accumulating, it is because there is a lysosomal storage disorder, meaning there is increased accumulation, there is increased storage of this particular substrate inside our cells. Okay, so these Niemann-Pick disorder people will be presenting to you with cherry red in the retinoscopy and hepatomegaly will be there. Okay, when we diagnose a patient with an Niemann-Pick disease and we are examining him, cherry red spot will be there in the retinoscopy and hepatomegaly will be there. Okay, so contraindicating to that, Tay-Sachs disease, here the GM2 ganglioside will be defect, GM2 ganglioside storage will be increased, where we will get to see only cherry red spots in the retinoscopy. So that is why please do not confuse name and whether. So they would give one cherry red spot. Definitely you will be thinking, oh, whether this is Niemann-Pick disease or Tay-Sachs disease. So that is when you have to see whether there is hepatomegaly that is given in the question or not. So if hepatomegaly is given, then it is going to be Niemann-Pick disease. If it is not given, it is Tay-Sachs disease. Then Gaucher's disease, this definitely you would have solved in all your question banks. Tissue paper crumbled appearance. Major deficiency is glucocerebrosidase deficiency. Okay, this is not the storage form, this is the deficiency of the enzyme that is there. Okay, glucocerebrosidase. Then when it comes to Fabry's disease, we'll see Maltese cross in the urine. Okay, that is all and everything that you need to know about lysosomal storage disorder. Then when it comes to the disorders of the fat, it is going to be called as dyslipidemia. All the metabolism that I explained to you with regards to fat metabolism and all, right? So let us see what are the defects that we can see here. So dyslipidemia can be classified as 4 types. Okay, type 1, type 2, type 3 and type 4. What is type 1? Type 1 is LPL receptor defect. What is LPL receptor defect? Remember, right? ApoC2 was detected by lipoprotein lipase, which converted it into triglycerides and fatty acids. Suppose if this lipoprotein lipase itself is absent or there is a defect in it, what happens? What will happen? The chylomicron that was formed will start accumulating in the body itself because there is no utilization by the liver. So that is when these chylomicrons and triglycerides starts increasing and there is the risk of pancreatitis. Okay, that is the type 1 dyslipidemia. Coming to type 2, it is gonna be LDL receptor defect. Okay, I told you, right? So in adipocytes there are LDL receptors. When the LDL receptors in the adipocytes are not there, then the LDL starts remaining in the blood itself. Right, in the peripheral tissue it is not getting utilized, so it starts remaining in the blood. So what happens is LDL and cholesterol starts accumulating in the blood itself. It will lead to something known as tendon xanthomas. Okay, in the tendons of our body, suppose it is the Achilles tendon, all these things, these fat blobs like these LDL blobs will start accumulating over there. In type 3, it is ApoE defect. ApoE, what was it doing? In both the chylomicron and VLDL, the remnants were being taken by the liver through recognition of ApoE. Suppose this remnant is not being taken up, then the chylomicron and the VLDL both gets accumulated. That is when we are gonna get to see the fat blebs that is getting stored in the palm. Okay, palmar xanthoma is what we can see. Then the type 4 is VLDL overproduction. I told you, right? VLDL overproduction, if it happens, then it is gonna be VLDL will be accumulating in the body, VLDL cholesterol, because LDL it will be increased. VLDL, if it is increasing, LDL definitely will be increased because from VLDL only LDL is forming, right? So both of them will be increased. So that is where we can also get to see pancreatitis. Okay, so please they'll give you the whether tendon xanthoma can be given, palmar xanthoma or pancreatitis can be given. So you should differentiate based on the laboratory values that they'll give it to you. Okay, this is the basic pathology or pathogenesis behind this dyslipidemia. I want you guys also to remember there's something called as A beta lipoproteinemia, where the MTP, that is microsomal triglyceride transfer protein deficiency is there. So there is no formation of chylomicron or VLDL. Okay, so here the fat absorption itself is not there. So I told you, right, the fat fat soluble vitamins, that is A, D, E, K. If the fat soluble vitamins are absent, then there is going to be oxidative stress. I told you, right? Vitamin E is very important in maintaining the antioxidant levels in the body. When the oxidative stress increases, we can see stress on RBCs and we can see acanthocytes. Okay, more about this in hematology. Okay, so I want you guys to remember, just because we were discussing about lipid metabolism, I also wanted you guys to remember A beta lipoproteinemia. That's it with respect to metabolism, guys. Okay, so now moving on to enzyme biochemistry, there are only three things that I want you guys to learn. One is, in enzyme biochemistry there is inhibition. Okay, enzymes basically inhibits or promotes the promotes the metabolism. Okay, so if there is an inhibition, there are three types of inhibition done by the enzymes. It is competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. Okay, so the graph that is gonna come will be X shaped in competitive inhibition. Suppose this is a graph that they'll give, and if it is an in X shaped, please remember, go and mark competitive. Okay, so in non-competitive, N. Okay, so it is just gonna look like N. Okay, so this is one graph. So this V shape is there, then it is gonna be non-competitive. Uncompetitive is U parallel. Okay, U is there, so two parallel things are there, right? See, these two parallel things. Okay, U, if it is there, then it is going to be uncompetitive inhibition. Okay, you need not know what is all this pathology behind this conversion and all, the inhibition and all. So they are more interested in asking what this graphs and what inhibition that particular graph is doing. So that is the identification of the graphs that they are asking. So one more thing is Vmax and Km value that they are asking. You need not know what is Vmax and Km also, just please remember this thing. Okay, so in competitive inhibition, you know Virat Kohli, right? So if Virat is consistent, then Virat is consistent, Kohli will score scores. Okay, Kohli is gonna score too much. Okay, so that is Vmax and Km. Vmax is consistent, then the Km value will be increased. Whereas if Virat is non-competent, then Virat Kohli is non-competent. That means Virat will be a low. I mean, with low his caliber, then he will consistently score ducks. We all have seen this, right? When Virat is low, is consistently scoring duck. But when Virat is consistent, he will be consistently scoring runs, hundreds, and all. But when it comes to uncompetitiveness, both Virat is low and his score is also low. Okay, when he is non-competent, his score, Kohli will be ducking. When Virat is consistent, his score will be high. So this is how you can remember. Okay, so this is a memorable way that you can do. Okay, and one more thing, important thing I missed to tell you with regards to hyperammonemia was, note that in hyperammonemia, if there is a GI bleeding, there is increased chances of ammonia synthesis to increase. Okay, so due to breakdown of all the materials that is happening because of GI bleed, there might be increased ammonia formation. Okay, hence try to treat the bleeding first. Okay, we'll discuss more about this when it comes to hepatology, because we can see the hepatic encephalopathy and all, right? So there ammonia is increased and all. So there the veins, that is due to portal hypertension, there is bleeding also, venous bleeding that will be occurring inside our gastrointestinal tract also. That is why we will have to treat our GI bleeding first and then go for treatment of hyperammonemia and other systemic treatments. So that is why we'll have to focus more on GI bleed when treating hyperammonemia. Please remember this. Okay, so there might be a question, they have asked it in some other exams, but not in Neetpg or INICET, but this is very much important. Okay, so that's it with respect to biochemistry, guys. You are done with biochemistry, all the metabolism and all. Just go through once and you will be done with biochemistry once for all. Okay, Thank you.